Hey guys, welcome back for chapter 10 notes. This will be on photosynthesis. So our world is solar powered. What that means is it's powered by the sun, really. All life on Earth gets their energy essentially from the sun or from light. That process starts with plants and other organisms that can do the process of photosynthesis. So if you remember from the first law of thermodynamics, sometimes we call plants producers, but in reality, they're just converting solar energy or light energy into chemical energy. That chemical energy would be in the form of sugars. And again, they are producers of the biosphere. In essence, photosynthesis nourishes the entire world, either directly through plants or indirectly through consumers, because all organisms use organic compounds for energy through cellular respiration. So plants are considered photoautotroph. Photo meaning light, autotroph meaning they have to they can produce their own energy. But the ultimate source of all organic compounds for heterotrophs is from these autotrophs. Autotrophs is equivalent to producers. Just again, prefix photo always means light. So photosynthesis, what we will focus on is plants. However, there are other organisms that can do photosynthesis, such as algae and some protists. Heterotrophs, that would be like us. We are considered consumers, which means we have to eat in order to get our energy. We have to feed on other organisms. Decomposers would also fall into this field. Decomposers do consume other things while they are breaking things down. But again, all heterotrophs depend on the photoautotroph for food and also for our oxygen. So photosynthesis, we are converting light energy into chemical energy of food. This can happen in all green parts of a plant. However, we'll look mostly at the leaves because that's the major site. That's where most of it happens. In 1 square millimeter of a leaf, you can find millions upon millions of chloroplasts. So it's pretty crazy to think about. Chlorophyll would be found in a chloroplast, and chlorophyll is a green pigment. It helps to absorb light energy, and most chlorophyll would be found in that mesophyll part. So if we look at this cross section, that's in the middle part. But really, if the plant is green, any green part probably has chlorophyll in it. As far as gas exchange, we have these little openings in the bottom side of a leaf. They're called stomata. Carbon dioxide comes in through there and oxygen would leave through there. Water, you can see, would come from the vein, eventually, usually taken in by the roots and then transported up through capillary action. Chloroplasts are the green organelles that do photosynthesis, and there's 2 real structures that we will concern ourselves with, and that is the thylakoid and the stroma. So the thylakoid is one of these green discs. A group of them is grana. That's where that term comes from. But as far as the stroma, when I think of stroma, I think of space. It's kind of almost equivalent to the cytoplasm of a cell. It's a very aqueous or watery filling of the chloroplast. So that space inside there is the stroma. So photosynthesis can be summarized as this equation. One thing you might notice on here is that water is on both sides. So a lot of times it'll get simplified and we won't include water on this side. We just kind of cancel those 6 out and we say there's only 6 going in. So a lot of times what you'll see is 6 carbon dioxide plus 6 water, and including light energy is going to yield glucose and 6 oxygen. So just to be aware of that. And really that would be the reverse of cellular respiration if you think about it. So chloroplasts are going to split water. That's one of the first steps. They're going to split water into its component parts, which is hydrogen and oxygen. And that would be why we release oxygen in photosynthesis. That comes directly from the splitting of water. But you can see where some of these other elements are going. The carbon that was going to go into glucose comes from carbon dioxide. That's a good one to know as well. Photosynthesis is also a redox reaction. Remember, oil rig, oxidation is loss, reduction is gain. What they're talking about is electrons in that. So in reduction, um, reduction is gain of electrons, and in oxidation, oxidation is loss of electrons. So in the photosynthesis process, water is oxidized, meaning water loses electrons, and carbon dioxide is reduced, meaning it gains electrons. And we'll go through a process of how that works. So just to preview the process, photosynthesis does consist of two processes, the light reactions and then the Calvin cycle. This would be the light reactions. They do occur in the thylakoids. Grana is just a group of thylakoids. So this is a thylakoid membrane right here. And what we do, the first step is to split water. That would happen right here. And when water splits, you can see there's oxygen and then hydrogen ions. But it lost electrons, and electrons would be boosted through here. This little squiggly yellow line is light coming in. So that electron would move through an electron transport chain, eventually get boosted again and make NADPH. Similar to NADH, easy way to distinguish between those, the P, the NADPH. Think of photosynthesis for that one, and you'll be able to remember that NADPH is for photosynthesis. And that is still an electron carrier. It does the same job as NADH and cellular respiration. So along this electron transport chain, one of the things it does, similarly to the electron transport chain in cellular respiration, it pumps hydrogen ions across the membrane. Okay, that's the same thing that the electron transport chain did in cellular respiration. It uses the electron's energy to do that from low to high. So we're going to have a high concentration of hydrogen ions in here. Eventually, they will move again through ATP synthase from high to low, diffusing through there, and that's going to create ATP. One of the things you'll notice is that the NADPH and the ATP will move on to the next cycle, or the next process, which is the Calvin cycle. So this is the light reaction. Why? Because it requires light. So the Calvin cycle is separate from that, and the Calvin cycle's job is to create the sugar. You'll see that carbon dioxide enters. There's going to be a series of intermediates here. You do not need to know those intermediates. We really focus on 3 steps, which is fixation, which is fixing carbon into sugar, reduction, which is adding ATP and NADPH is energy into that sugar, and then regeneration, which is just recreating the molecule that is going to be able to accept more CO2. So that's 3 steps that we will focus on this week in class. So again, light reactions require light. Water is going to go into there. Oxygen is going to be produced along with ATP and NADPH. Then the Calvin cycle, CO2 is used along with, it's going to use the ATP and NADPH. And then so you can see that cycle, that connection between the two is the ATP and the NADPH. And the Calvin cycle's job is to create that sugar. If you want to review that overall process, this is a good video to do so. And you have these slides, so you can do that at any time. I'm going to move on into the light reaction just to review here. So light is a form of electromagnetic energy. It travels in waves. It travels in photons, which is a particle of light, which is on your reading guide, I know. So thylakoids are going to convert light energy into chemical energy of ATP and NADPH. A wavelength is simply the distance between a crest of a wave. So if I were to draw that quickly, if this is a wavelength would be, this would be a crest and this is a crest. So this distance between them would be a wavelength. So the electromagnetic spectrum covers all of this, but really what we use for photosynthesis is considered the visible light spectrum. It is definitely the most important band of light for life. The atmosphere screens out a lot of radiation, which would be the stuff that doesn't fall into this. The visible light is only the only form that passes through in significant quantities through our atmosphere. So the visible light spectrum includes all the colors of light that we can see, and includes the wavelengths of light that will drive photosynthesis. We'll talk more about that this week. A photon, again, is a particle of light. Shorter the wavelength, the higher the energy. Pigments are going to be substances that absorb light. So chlorophyll is an example of one. It is a green pigment that absorbs light, except for green. It will reflect light. That is green. You're seeing that here. So the light is coming in, which would be all colors of light. Green gets reflected. So if it's reflected, remember it's not being used, because if it's reflecting it, it's not using it. So it's going to use all colors of light except green. How we measure that would be through a spectrophotometer, which is just a device that is going to help us to send light through certain pigments, and we can measure how much of that is getting absorbed versus reflected. So you can see here we're sending light, and we're going to separate that light into colors through a prism, and then you're just sending different colors of light through. So this one is green, and they're measuring how much light is passing through. So you can see on this meter over here, a lot of the light is passing through. Very little is being absorbed, in other words. If we look down here, this one is blue light, and very little of it is passing through. So most of it is being absorbed, in other words. So that's what we're measuring there, that we're measuring how much gets absorbed. So the absorption spectrum of chloroplast pigments is very interesting. This is a concept that is on the AP biology test quite often. It will be on your unit test for sure. But it provides clues to the relative effectiveness of different wavelengths that drive photosynthesis. You'll be doing a lab and pivot this week that will also measure this. But chlorophyll A is the dominant pigment that absorbs the best in red. It absorbs best in the red and the violet to blue range, and it definitely absorbs green the worst. So you can see that here. Here's our chlorophyll A. Here we have violet and blue light, and you can see it's relatively high, versus down here in the green section, very, very low, and then it peaks again in that red. And that really is associated with all of the different pigments that are associated with photosynthesis, which would be chlorophyll A, chlorophyll B, and carotenoids. Those are really the three pigments that photosynthesis uses, and all of them use the same really types of colors of light. So green is definitely not used. Yellow is also another one that's not really well used, hence why we see them as green, because most of that light is being reflected. If you think about it, if it's being reflected, we can't really use it for the process. So this is another way we can measure photosynthesis. In this case, what we're measuring is oxygen. And if we put this, they don't have a x-axis here, but if we put the light spectrum here, green, you could predict would be down in here. This would be like blue and red, violet again, and this would be red. So chlorophyll A is that main photosynthetic pigment. We do have accessory pigments like chlorophyll B and carotenoids. So when a pigment absorbs light, it gets excited, and what it really does is it excites electrons, and we'll show you that in the photosystems in a minute here. Photosynthesis does a really nice job of harnessing. If we don't do this process in the leaves, in the photosynthetic leaves, if you just had chlorophyll like this in a beaker, it actually gives off a lot of light and heat, but we can harness that light in photosynthesis. This is raw chlorophyll. This is not chlorophyll that is found in a plant cell, in a chloroplast. So here we have it this is a membrane of a thylakoid. This would be a photosystem. So we have light coming in, and what you're seeing here is it's being funneled. Here is our chlorophyll A. So these would be like our accessory pigments, chlorophyll B, carotenoids. Well, again, what are they doing? They're funneling their light to chlorophyll A, which is like the reaction center, and they're going to boost an electron. What that means is it's boosting the energy of the electron. So the light harvesting complex is going to consist of pigment molecules bound to particular proteins. That would be the photosystems. So the reaction center, again, it's going to be that primary electron acceptor. Chlorophyll is going to accept those electrons. Where do those electrons come from? It's really water. So thylakoid membrane is populated by two types of photosystems, photosystem 1 and photosystem 2. So step 1 has to be light entering those photosystems. And I'm going to kind of skip ahead a little bit quickly here. And the reason why is I want to get to the diagram so that I can show you this. But non-cyclic electron flow, this is the primary pathway. This is normal, the normal pathway. And the end goal of this is going to use light to create ATP and NADPH. So here is a kind of a model of the whole process, but here's photosystem 2. So photosystem 2 is going to have light come in. We're going to funnel it to the chlorophyll A, which is going to boost an electron. Where did that electron come from? Here it is, from water. So we're going to use the light's energy to split water, and that's where your oxygen comes from, as well as some hydrogen ions. Boost that electron, and it goes to the electron transport chain. Electron transport chain's main job is going to be to create ATP. In photosystem 1, again, light will come in, we'll funnel it to chlorophyll A, boost that electron again, and over here we're going to create NADPH. So again, those are the two main things that we're going to create is ATP and NADPH. These are light reactions. How do I know that? Because we're using light. I'm going to skip ahead a little bit here. Cyclic electron flow is a little bit different. The reason why we have cyclic electron flow is simply because in the Calvin cycle, we use ATP more than we use NADPH. Because of that, we need a mechanism in order to create a little bit more ATP at certain times. So what happens is rather than dropping it off this way in photosystem 1 and creating NADPH, which would be normal, sometimes we can kick it back to the ETC and have, so you can see why it's cyclical. And because of that, we're going to create a little bit more ATP. So it's just a mechanism that plants have to create more ATP. The only reason, again, they have this is the Calvin cycle uses more ATP than it does NADPH. Normal electron flow or non-cyclic would create equal amounts roughly of ATP and NADPH. So chloroplasts and mitochondria are going to really generate ATP, pretty much the same mechanism. They both use chemiosmosis, which remember is the creation of that gradient, eventual diffusion of those hydrogen ions through ATP synthase to create ATP. They use different sources of energy, right? Because the light is the source of the energy in photosynthesis for chloroplasts, but in a mitochondria, we're going to use sugars. So in that way, they're different, but the mechanism really looks the same. So you're going to have hydrogen ions diffusing through ATP synthase to create ATP. We're going to use an electron transport chain to create that gradient. So that process is really, really similar. Both organelles do use redox reactions. They're going to use electron transport chains to generate that gradient, that hydrogen ion gradient across the membrane, and then they're going to use that ATP synthase to move them back, to diffuse them down their concentration gradient and create ATP. So here's that overall process, and we will review this extensively in the next week or so. Definitely, if you're a robust virtual student, you're going to want to join class this week. If you missed the Monday, Tuesday version of that, send me an email and I'll get you an invite for Thursday, Friday, because it'll be really good for you guys to get some good review with me, reviewing with you on these concepts. So this is the light reaction. I'm just going to review that again. Really what we're seeing here at step 1 is water is coming in and being split. We're going to use the light's energy in order to split water. And what that is doing is it's going to split into oxygen, which is why oxygen is a byproduct of photosynthesis. The oxygen that you're breathing right now, it came from this process right here. The other thing when we split water is we're going to create some hydrogen ions. And that's going to, again, see that it's going to be in the thylakoid space, in the inside. Meanwhile, electrons were boosted from that split water. The light's going to energize them. They're going to go down an electron transport chain. So at number 2 here, what you're seeing is we're going to use that electron's energy to pump more hydrogen ions inside the thylakoid. So those hydrogen ions that are inside the thylakoid came from both the splitting of water and the electron transport chain. Eventually, that electron does get to our third step, which is at photosystem 1. More light is going to come in. We're going to boost that electron again, and it's going to go to a second electron transport chain where its job is to create NADPH. Okay. Then the last part of this is we're going to use that huge high concentration of hydrogen ions. They're going to diffuse down their concentration gradient through ATP synthase to create ATP. You'll notice that both NADPH and ATP go on to the Calvin cycle, which is next. The Calvin cycle's job is to create sugar from CO2. It's also going to use that ATP and NADPH that we just talked about that was made in the light reactions. The Calvin cycle is somewhat similar to the citric acid cycle. It also occurs in the stroma, which remember is that space inside of the thylakoid, kind of similar to the cytoplasm of a cell. It occurs in 3 phases. We'll go through each: carbon fixation, reduction, and then the regeneration of the molecule that will accept CO2. So here we go. The first step is fixation, and what that means is we're going to fix carbon dioxide into sugars. So Rubisco is an enzyme that aids in that process. You do not need to know Rubisco or any of these intermediates. What you do need to know is that CO2 gets fixed or combined into sugars. That's carbon fixation. The next step is reduction. And in reduction, we're going to use ATP and NADPH to make a G3P. Now, you might say, well, Mr. Tepes, didn't we create glucose? And you'd be correct. 2 G3Ps would create a glucose. Okay, so you might understand why. You can see there's 3 CO2s that go in, 3 carbons there, right? So this would be a 3-carbon sugar that we create. So it would take 2 cycles of the Calvin cycle to create a glucose. 2 G3Ps would create a glucose. All right. So that's what happens in phase 2, reduction. The third phase, we're going to use the remaining ATP to regenerate the molecule that will accept more CO2 in. In other words, allowing this cycle to continue. Now, remember I said not cyclic electron flow is a thing because we use more ATP. Well, let's count them. We got 3 there and 6 here. That's 9 ATP are used. 6 NADPH are used. So that's, again, why cyclic is a thing, because we use more ATP than NADPH. So just to review the light reactions, we're going to use light and water. We're going to give off oxygen, and that light reaction's job is to create ATP and NADPH, which are used in the Calvin cycle. The Calvin cycle is going to use CO2. It's going to create sugars, and it uses that ATP and NADPH, and then since you can see they are directly connected in that way, through the ATP and the NADPH. This week we will watch this. If you would like to watch it at any time, you can definitely feel free. It's about a 10-minute video, but it does a really nice job of breaking that into steps. So we will definitely be watching that this week in class. But maybe if you missed class, if you're a robust virtual and you couldn't make any of the live options, or if you just want to review, this is a great video for review of those 2 processes. And that's it for photosynthesis. Again, if you are a robust virtual student, if at all possible, please try to make it in for live class this week.